So for fermentation is actually a way that allows us to regenerate all of the things we need for glycolysis. So if we can't, the schematic shows if we have glucose, we go through glycolysis to get pyruvate. If there's no oxygen, we can't shuttle everything we produce in glycolysis into the mitochondria because we don't have a final acceptor. So if that's the case, we will do fermentation. If we do have oxygen, obviously we want to get the most ATP possible. So then we would go through all of cell respiration. But in the case that there's no oxygen available, then your cells will do fermentation. And there's two types. We mentioned this in class on Monday, that we can do lactic acid fermentation, which is what we use. And then certain organisms can do alcohol fermentation. Anybody know which organism does alcohol fermentation? Yeast, right? So that's obviously how we get bread
Beer.
and beer and wine, right? So fermentation. So for the blank here, fermentation is used to regenerate. Specifically, it's used to regenerate NAD plus, so that we can continuously do glycolysis. Because as you might realize, we have to input that NAD plus to produce NADH, and if we don't have a source, we can no longer do glycolysis. So what this looks like on the energy diagram, the green section here is showing glycolysis. It's showing chemical potential energy of glucose. Remember, we invest ATP, so the first two steps raise the potential energy, and then the later steps harvest some energy. We get down to pyruvate, and if we don't have oxygen, we need to actually take pyruvate and reduce it to produce either ethanol or lactic acid. And that allows us to replenish the NAD+. I'll show you the chemical reaction for that. So first let's talk about lactic acid fermentation. This again is kind of showing you the schematic. This is glucose being converted into pyruvate. That's 10 steps of glycolysis. This process required NAD+ and picked up electrons, so it produced NADH. When we take pyruvate and convert it into lactic acid, the first question is this a reduction or an oxidation? So comparing lactic acid and pyruvate, which one's reduced? Yeah, it is a reduction. So you can see here we have double bonded oxygens. Here we have more hydrogen, so this one's reduced. So this is a reduction. And if we're going to reduce it, we need something to be the source of electrons. So what's the source of the electrons? Is the NADH. Passes its electrons to pyruvate. That gives us lactic acid. Okay, so that regenerates our NAD+. And again, that was the goal for that. For alcohol fermentation, we're also going to start with pyruvate. But the first step is to take this carboxyl group and remove it. So obviously when we have ethanol fermentation, we produce CO2. So if you think about beer production, right, you have CO2 in beer. The CO2 is a byproduct here, and then you produce acetaldehyde. The acetaldehyde is actually what is then going to be reduced. So again, it's picking electrons from here, NADH. That's giving us our starting NAD+, and that makes the reduced ethanol from acetaldehyde. Okay? So, does anyone have questions on those two different types of fermentation? The purpose, again, is just we need NAD+ if we're going to repeatedly go through glycolysis. If we are only performing fermentation, how many ATP are we producing? Just the two, right? We can't even get into the TCA cycle or anything like that. So we're really limited in how much ATP we make, but at least we're making a little bit. Okay. All right, so what happens to that lactic acid? I think probably many of you have heard of like lactic acid causing muscle soreness. I think this is believed to actually be more of a myth, that it's not the buildup of lactic acid that causes muscle soreness, But what happens with the lactic acid after you produce it? So this, again, I took this from a nutrition textbook, but showing if you have glucose being converted to pyruvate, and before we have oxygen delivered to the muscles, we will then use fermentation and produce lactate. But we can actually recycle the lactate. So lactate goes to your liver, and there's a process that converts it back into glucose, which means that it ultimately is used as a fuel source. The process of converting lactic acid or lactate into glucose is called the Cori cycle. So I think in biochem they do briefly talk about this process in your liver. Your liver is doing so many different metabolic reactions, but that's one of the things that it does. It ultimately gets recycled back to glucose, and then it can be used as a fuel source. Okay, so the last slide that I have comes back to this one, which we looked at the beginning of this lecture. And first I just showed you this, right? This is our carb metabolism pathway that we talk about. But I made the point that part of the reason we do this is to give us all of the macromolecules or the intermediates that we need for these other pathways. We use a lot of the intermediates, to make nucleic acid, to make amino acids, to make our lipids. So it's not only important to produce ATP, but also gives us these other components. And then just so we kind of understand how our food filters into this, we don't just eat glucose, right? We eat lipids, we eat proteins, we eat carbs, simple and complex carbs. Most of the carbohydrates that we consume get broken down into glucose or fructose or one of the other sugars and enter in through the glycolysis pathway. Our fats, if we're taking in triglycerides, they're getting broken down into glycerol and fatty acids. The glycerol gets converted into pyruvate, so it can enter in through the glycolysis pathway. Fatty acids get processed 2 by 2, 2 carbons at a time to make acetyl-CoA. So these filter in through this point of the pathway. And then our proteins that we consume depends on the amino acids. Some of them enter in through, they get converted to pyruvate, and some of them get converted to intermediates in the citric acid cycle. So that's how they would enter into this main carb metabolism pathway. There's 2 different breakdowns for the foods that we eat. They're considered glucogenic or ketogenic. If they are glucogenic, they are compounds that can be converted into pyruvate, or they enter in through the glycolysis pathway. So the definition I wrote down for glucogenic is they're compounds that can be converted to glucose or somewhere along the glycolysis pathway. They enter in through here. So for example, carbohydrates are called glucogenic. Glycerol is a glucogenic compound. And some amino acids are considered glucogenic. You don't need to worry about which ones. But for ketogenic, these are compounds that can be converted into acetyl-CoA or into intermediates in the citric acid cycle. And so they include things like fatty acids, those are called ketogenic, and some proteins, certain amino acids are ketogenic compounds. Okay, but basically everything that we eat has a way to get into this main metabolic pathway. All right, so any questions on cell respiration, or everybody feel solid on that topic? Okay, so now we're going to shift. Our very last pathway is actually not too long because I've shaved it down over the years. This is photosynthesis. So we're going to talk about photosynthesis, which sort of fits in with sort of the opposite of cell respiration. But this is a practice exam question I had in here. So if you want to read this, so infants and animals that do not shiver, they have this special tissue called brown adipose tissue as a means for thermal regulation. And so this brown adipose tissue contains uncoupling proteins that render the inner mitochondrial membrane permeable to protons. What would be the consequence of allowing protons to pass freely through the mitochondrial membrane? And so again, this is kind of related to cell respiration. But think about that diet drug that we talked about. And if infants are needing thermal regulation, how could this uncoupling protein help to give them that thermal regulation? So with the people next to you, just chat about this for a second, and we'll see if we can come up with how this brown fat works.